Good morning, students. I am Dr. Harsha Shodhi from HUHP Institute of Postgraduate Studies and Research. Topic of my today's discussion is microbial interactions. This is the second lecture of the same topic. The topic is a part of BSc Semester 5, CMB 501, Environmental Microbiology. The reference books for this are Microbial Ecology by Atlas and Bartha and Press Microbiology by Prescott. In the first lecture, we saw the definition of ecology. We saw what are interactions. Interactions are of two types, positive interactions and negative interaction. An interaction in which one or both the species which are participating in an association are positively benefited in terms of food, shelter, substratum or transport is termed as a positive interaction. Interaction in which one or both the species which are participating are negatively affected in terms of food, resource, space is termed as a negative interaction. Positive interaction are of four types: mutualism, commensalism, proto-cooperation and neutralism. An association in which both the species are benefited and it is an obligate association, that is a compulsory association, is termed as mutualism. We saw the detailed discussion, or we had a detailed discussion of mutualism in the last session. Today we'll start with commensalism. Commensalism is an association in which both the species are positively benefited. But this association is not a compulsory association. In absence of the association, both the species can survive. Commensalism in Latin, com means together and mensa is a table. It is a relationship in which one symbiont, that is the commensal, is benefited, but the other participating species, which is called as host, is neither benefited nor it is harmed. This is very important. The host should not be harmed. Otherwise, if the host is being harmed, the association goes into a negative interaction. Commensalism describes the relationship in which one organism lives off the table scraps of other. It is a charity. It is like a charity when one population contributes to the charity or gives something which the other population is taking up. So the host is the person who will contribute to the charity, and commensal is the person who will take the benefit of that charity. It is not an obligatory interaction in which one population benefits while other remains unaffected. It is an unidirectional relationship between the two populations. The unaffected population will not benefit, but at the same time it is not negatively affected by the action of the second population. The recipient population will need the benefit which is provided by the host. But as I said, this is not a compulsory or an obligate relationship. The same thing may be possible if the commensal will receive the same assistance from another population with a similar metabolic capacity. That means if one partner is not ready to contribute to the charity, some other partner can contribute of a similar metabolic capacity. The spatial proximity of two partners permits the commensal to feed on the substances which are captured or ingested by the host, and the commensal will often obtain shelter by living on or in the host. Please see, when the commensal is in an association of commensalism, both the partners are living in a close proximity. So the commensal will feed on the substance which is given out by the host. At the same time, it can obtain the shelter, but it is not obligate or compulsory. The commensal is not directly dependent on the host metabolically, and it will not cause any harm to the host. There are a number of physical and chemical bases for the relationship of commensalism. Commensalism is observed in case of habitat, it is observed in case of growth factors, it is observed in case of modification of the environment. For example, we'll first see habitat modification. Commensalism in the habitat is observed when the unaffected population in its normal growth and metabolism will modify the habitat in such a way that the another population will benefit because of the modified habitat, as the modified habitat is more suitable to the need of the organism. A simple example is that consider an environment which is having all aerobes, facultative aerobes, and anaerobes, and there is presence of oxygen. In presence of oxygen, obligate anaerobes cannot grow. But when facultative aerobic organism or aerobic organisms will grow, they will utilize the oxygen in this place. When they use up all the oxygen, the habitat becomes anaerobic. So what has happened? By the growth and metabolic activity of aerobic and facultative anaerobic organisms, an aerobic environment has become anaerobic, which is now suitable for the growth of obligate anaerobes. So in this association, obligate anaerobes are the commensals. They are being benefited by the growth and metabolic activity of facultative aerobes and anaerobes who use up the oxygen, and they will modify the environment such that obligate anaerobes can survive. So obligate anaerobes get the advantage from facultative aerobes and anaerobes, and facultative aerobes and anaerobes are having no loss in this. It is only during their normal growth and metabolism they have modified the habitat such that it becomes more suitable for the obligate anaerobes to grow. This example is discussed over here. When the population of facultative anaerobes use oxygen, they will lower the oxygen tension. This will create a habitat which is more suitable for the growth of obligate anaerobes. In such a habitat, obligate anaerobes have benefited from the metabolic activities of the facultative organisms. The facultative organisms at the same time were unaffected by the relationship, but this will be true only if the two populations are not competing for the same substrate. That means they should not be competing for the same carbon source or nitrogen source. If they compete for the same carbon source, the association will not remain as commensalism. It will shift towards competition. So this will be true only and only if the two populations are not using the same resources. The occurrence of the obligate anaerobes within the microenvironments of predominantly aerobic bacteria is dependent upon the commensalistic relationship of aerobes and the obligate anaerobes. A second example which we see in commensalism is production of growth factors. Many organisms, when they grow, during their growth and metabolic activity, they excrete certain substances. This excreted substances may be growth factors which will be required by the other organisms. The production of growth factor forms the basis of commensalistic relationship between the microbial population. Many microbial population produce growth factors like amino acids, vitamins, etc. This type of growth factors can be used by other microorganisms of the population. For example, Flavorbacterium brevis, it excretes cysteine. This cysteine is a sulfur-containing amino acid, which is used by Legionella pneumophila in an aquatic habitat. So Flavorbacterium is the host, which excretes cysteine in its normal metabolic capacity. This cysteine is a growth factor for Legionella. So Legionella is a commensal which will be using up the cysteine. In soil habitats too, we find many bacteria which are dependent on the production of vitamins by other microbes. The production of growth factors and their excretion allows fastidious microorganisms to develop in the natural habitats. A very interesting example I would like to quote over here. It is said that we can only culture 1% of the total microbial population which is there. 99% of the microbial population which exists is still not cultured. It is thought that we cannot culture this microbial population because in nature there are a number of growth factors which we cannot mimic. So where does this all come from? This all comes from the interrelationships of the microorganisms. They're providing them with growth factors which are unidentified, which we cannot replicate or mimic in the laboratory. As a result of this, we have not been able to cultivate still 99% of the microbial population, which is still unknown to us. So it may be that a commensalistic relationship is occurring, which we have not yet not deciphered. Then we come to transformation. A third type of commensalism is a transformation, where transformation is of insoluble compounds to soluble compounds, conversion of soluble compound to the gaseous compounds, which forms the basis of commensalistic relationship. For example, methane is produced in anaerobic sediment. This methane can be used by the methane oxidizing bacteria in the above sediment. So this becomes a commensalistic relationship, which is explained over here. Methane, which is produced by the bacterial population in the sediment, it can be used by the methane oxidizing population in the overlying water column. So methanogenesis results from the commensalistic relationship for the population. The example quoted over here is of D-sulfovibrio and methanobacterium. D-sulfovibrio can supply methanobacterium with acetate and hydrogen. This acetate and hydrogen comes from the anaerobic respiration and fermentation when D-sulfovibrio uses sulfate and lactate. So D-sulfovibrio will use sulfate and lactate in anaerobic respiration. Its byproduct will be acetate and hydrogen. This acetate and hydrogen, which are produced by D-sulfovibrio, can be used by methanobacterium to generate methane. So what has methanobacterium done? It will use the product of D-sulfovibrio and it will reduce CO2 to methane. So this is a commensalistic relationship whereby D-sulfovibrio will use sulfate and lactate, will produce acetate and hydrogen, and methanobacterium will convert CO2 to produce methane. The activities of one microbial population will also make a compound which is available to another population without actually transforming a particular compound. For example, number of bacteria are known to produce organic acids. This acids will release compounds which are actually insoluble and make it available to the second population. Such a desorption process are found in the soil, where many compounds are bound to the mineral particles or humic particles, which can be made available to the other populations, which will grow and metabolize using this compounds. Another basis of commensalism is conversion of organic molecule by one population into a substrate for another population. A very good example for this is polymer degradation. We all know that cellulose is the most abundant polymer, but cellulose degradation requires a group of enzymes which are called as cellulases. When cellulases act on cellulose, the byproduct will be glucose. Now, cellulose as such is a polymer. It cannot be degraded by all compounds, but certain fungi can produce cellulases. When they produce cellulases, cellulose will be broken down into glucose or smaller particles, polymer, smaller carbon compounds, which can then be utilized by the other organisms. This example is explained over here. Some fungi produce extracellular enzymes which convert complex polymeric substances such as cellulose into simpler compounds such as glucose. These compounds can be utilized by other microorganisms of other populations which do not possess enzymes which can break down polymeric substances. So over here, fungi is the host. It will produce cellulases which will break down cellulose. The breakdown product of cellulose will be glucose. This glucose will be used by another population of microorganisms, which is commensal, which does not have the enzyme to break down cellulose. So again, if you observe here, this becomes a sort of succession where polymeric substances will be degraded. Degradation product of this polymer becomes a food for a second group of organism. When this second group of organism will use up this food, it will produce another metabolic product which can become a food for third group of organisms. So this relationship is commensalism, and it will also result into a succession within a closed environment. Then you have another basis of commensalism, which is called as detoxification, where organisms remove or neutralize a toxic material. For example, oxidation of H2S or hydrogen sulfide by Bagiatova. This is an example of detoxification. This will be benefited to those organisms who are sensitive to H2S in aerobic environment. Bagiatova does not benefit from this association. So Bagiatova will use up H2S. H2S is toxic to many organisms. When Bagiatova will use up H2S, it is using it for its own metabolic capacity, and it will detoxify the environment. Precipitation of heavy metals such as mercury by sulfate reduces sulfate reduction provides an additional example of detoxification. Some microorganisms detoxify the compound by immobilization. For example, leptothics group of organisms will reduce the manganese concentration in the habitat. This will make the environment favorable for that microbial population to whom this manganese concentration was toxic. So leptothics has used up manganese and in turn made the environment favorable for other group of organisms. And another example for this is co-metabolism. Co-metabolism is a very interesting example. Co-metabolism is largely applied. This is a very important association. This association has been exploited for bioremediation. We'll see what is co-metabolism. Co-metabolism is an association whereby organism grows on a particular substrate. The organism which grows on this substrate will oxidize a second substrate which it is unable to use as a nutrient and an energy source. In co-metabolism, again understand there are two organisms because what we are studying is a part of commensalism. So two species are being participating in this. One species is not being affected, one is being positively affected. Co-metabolism will have two species. Out of two species, there is one species which metabolizes a substrate for its growth. When it metabolizes one substrate for its growth, it will also metabolize or it will also oxidize a second substrate. The second substrate is not used for its growth and the metabolic capability or utilization. But when the second substrate is oxidized, it will be converted to a compound which can be utilized by another group of organisms. So again, coming back, commensalism is an association whereby organism grows on a particular substrate, it oxidizes a second substrate which it itself is unable to use as nutrient and an energy source. According to strict definition of commensalism, second substrate will not be assimilated by the host or the primary organism, but the oxidation product of this is available for use by another microbial population. I'll show you an example over here which will make it more clear. You can see over here, propane is converted by microbial or is metabolized by microbial vacuum. When propane is being utilized by mycobacterium, it also oxidizes cyclohexane to cyclohexanol. Now cyclohexane is converted to cyclohexanol. Cyclohexanol can now be utilized by Pseudomonas. Pseudomonas was unable to use cyclohexane, but it can use cyclohexanol. Again, going back to or explaining this, Propane is metabolized by mycobacteria for its growth and metabolism. During oxidation of propane, mycobacteria will also oxidize cyclohexane to cyclohexanol. Cyclohexanol can be utilized by Pseudomonas. It will be converted to cyclohexanol and will be further converted. So cyclohexanol is a substrate or which can be utilized by Pseudomonas, but Pseudomonas cannot utilize cyclohexane. So during the growth and metabolism of propane, cyclohexane was converted by mycobacteria to cyclohexanol. This cyclohexanol can now be utilized by Pseudomonas, which could not have utilized cyclohexane. So in this association, Pseudomonas is the commensal, mycobacteria is the host,